Well, hello, it's Miss Dagenford, and I wanted to put this video up more for your benefit if you wanted to review. I know we went over this on Friday, so there aren't any questions associated with it, but in case you needed to review it, here it is. So we started the other day with the kidneys, and kidneys are the organs for mammals and birds and reptiles that filter blood in order to remove nitrogenous waste, amphibians as well. Um, and then they also play a role in controlling the blood solutes, the water concentration and the solutes in the blood. So one thing you need to understand is that when we're metabolizing proteins, when we're just going through metabolic pathways, we produce ammonia, and that is very toxic. Our body is going to convert it to urea, less toxic. In the case of birds, it would convert it to uric acid, and reptiles as well. But we're going to look at urea. Now, the goal of the kidneys, and I think most of you know from just other classes, the goal of the kidneys is to filter the blood. And what you have, this is the outer cortex of a kidney, this area right here. And then down here we have the medulla. And what your body is constantly doing is sending blood into these tubes called nephrons. So this right here is a nephron. Now we have nephrons, some of which go all the way down into the medulla, and then we have ones that are very short. Now we want to look at the ones that are very long because they have the most interesting case study for what we've been looking at, diffusion and osmosis. So the blood to be filtered comes up and is going to be pushed into this little area called the glomerulus. And the glomerulus is just little cup-shaped little organ that allows the blood plasma to filter through. You want to imagine if you're cleaning your room and you decide to take everything out but the furniture. The big stuff stays, but you're going to throw the magazines or your books, notebooks, your CDs, your clothes. You're going to put them all out in the hallway, and then including the trash that you might have, and then you successively bring back everything that you want. So you're going to take your clothes back in, the magazines that you want, the CDs, and then you're going to keep the trash out in the hallway. So that's kind of what's happening here. Everything kind of gets pushed out of the blood except for the blood cells. And then the first stage is that you're going to reabsorb the materials that you want to reabsorb. And then what the kidneys can also do is help control things. So they can control pH and salt and electrolytes and all sorts of things, glucose. But primarily you want to manipulate the amount of water and urea that leave the body. Now you want to think that if we've removed everything, that includes water. And if you follow the pathway of the tube, that pathway is eventually going to go outside the body. This goes to the collecting duct, which goes to the ureters, which will eventually leave the body. Now that means water potentially leaves the body. Well, we don't want that. The kidneys process a lot of blood plasma. So the last thing we want is to lose water. And especially be thinking about the desert animals like kangaroo rats and camels and little reptiles. How do they handle even more of a challenge to not lose water? So here it is in a larger scale. So you can follow the arrows. The blood goes into the glomerulus, and the Bowman's capsule, and then this tangle of the blood vessels, the glomerulus, push everything out by the force of the blood pressure, except for the red blood cells. The filtrate goes through these tubules, and then this nephron dips down into the loop of Henle. and we have a descending limb and an ascending limb. And through this, there's going to be something very interesting. What we want to do is actually create a concentration gradient. So the area up on top has a much higher water potential. So it is less concentrated. It is more dilute. So up here it's less concentrated, and then down here it's more concentrated. And we're going to go over some different units that you might encounter. So this is much saltier down at the bottom, and then up top it's much more dilute. Now let's use our water potential terms. What ends up happening is up here we might have, and I'm just going to throw out some numbers here. I'm not worried about whether or not they're precise. The goal for you is to track the direction of the movement. So let's say in the little collecting duct here, as the urine is going to leave, let's say the water potential is -1. Well, as it passes down, let's say it encounters an area that's -8. So remember the dictum that we have for water potential. Water moves from an area of high water potential inside this tube to an area outside that is a low water potential. And let's just say, for the sake of argument, that's -8. So water is going to leave this tubule and go back to the body. So these are the body cells, and then the body capillaries. That's what we want. We want to be able to capture that water back. So as it leaves, it's not going to we're not going to lose that much water. Now in class, we did a little practice with this just by shading in, and we watched the video. What we end up having is something called the counter current multiplier, where, and it's part of this loop of Henle. And so let me write that down, counter current multiplier. And it's so called because the two loops, the descending limb and the ascending limb, so they run counter to each other. And then what happens is it multiplies this concentration difference. And it really starts coming to this concentration difference because of this active transport of sodium. So you can follow what happens. This is active transport. Sodium is going to be actively transported. This makes this area surrounding it much more concentrated with solutes. If you look at the other descending loop, that means water will passively flow out in response to that. So that's where you kind of get this multiplication. So as water leaves, it gets more concentrated. So it gets saltier and saltier as we go down here. And depending on how dehydrated you are, this will actually change based on that, because what we want is to create a concentration difference. Ultimately, that's the job of the loop of Henle, is to just establish a concentration difference. That really is it. Because a lot of students will look at this and go, well, what in the world did that accomplish? The filtrate goes down, gets saltier, and then as it comes up, because the sodium is pumped out, it gets more dilute. So then they look at me as if, well, that seemed useless. And, well, they're kind of right. What in the world is the point of that? Except you got to remember, the urine is taking one last turn as it leaves outside. So again, imagine if we're at the concentration, what they use is milliosmoles, and so it might be 300 up here, which is the typical for our blood. And as it goes down, it's approaching different areas that are more and more negative water potential. So this might be 300, but then the surrounding area is going to be like 600, and then down here it might be 800, and then as it leaves it might encounter 1200. So we're going from an area that has a high water potential to an area of low water potential, and that's what we're having the water leave, and we're recapturing it. And that's the important thing. Now, suppose, this diagram kind of maps out the numbers for us, which are really nice. So notice we're going to get this difference, and as it goes down, as the water's leaving, we're going to be able to encounter those concentration differences, and that's where the water leaves. Now, suppose we're very hydrated. If we're very hydrated, what we want is the urine to be very dilute, because think about it. If we're very hydrated, we've got plenty of water, we don't need to reabsorb it. So there's actually very little reabsorption taking place. Now, if we're very dehydrated, the opposite is going to be true. If we're very dehydrated, we want the urine to be very concentrated. In other words, very little water. Now, in order to accomplish that, we need to absorb a lot of water. We need to reabsorb back into the body tissues. We're going to reabsorb that water. And so now you know this experientially, because if you drink a lot, your urine is going to be quite almost clear. It's going to be very a little yellow color in it. But if you've gone without drinking a lot of water, your urine gets successively yellower and yellower. So if we just sneak down here, here's how it happens. Now, this should look, there's something in this diagram that should look a little familiar to you. You have these little vesicles that contain a molecule called aquaporin. And aquaporin is going to be really intriguing. It's this protein that really just creates pores, channels for water. Now, imagine up here, this is going to be our part A. So imagine this is a cell that has no aquaporins. There are no aquaporins in this section. If there are no aquaporins in this section, then think about what happens. Water comes down, and there's no passageway for it. There's no way for water to get in. So in this case, because there's no aquaporins, there's very little water absorbed back in. Because in this diagram, this over here, this is going to be our body tissues. This is going to go outside the body. This is essentially the urine. So think about what that means. Very little water reabsorbed. This is going to be the case where you are very hydrated. Now down here, imagine this is like our part B. So if we had drawn a different cell, part B, so notice these vesicles are fusing with the membrane through exocytosis, and now the membrane has plenty of aquaporins. Now, because they have plenty of aquaporins, follow the logic. If there's plenty of aquaporins, now we suddenly have channels to allow the water to go into the cell and then conversely into the body. So here we're going to absorb quite a bit of water. And this is the case where you would be dehydrated. So that gives you a little example of how our body at a cellular level can control things. And then we're going to look in a second how different animals can control or have different shaped kidneys to allow for it. And then at the bottom of the next page, we also have a different picture that just shows the number of aquaporins. And again, this is if we do not need to reabsorb it. So if we're hydrated, this bottom picture is if we're dehydrated, we want to absorb it. Well, hope that helps.